Hello, grade 12 students. In this video, we're going to talk about the hypothalamo-pituitary axis and ovarian secretions. So, document 3 of chapter 15, the hypothalamo-pituitary axis and ovarian secretions. First of all, you have to know what is the hypothalamus. Hypothalamus is situated at the base of the brain, just above the pituitary gland. This is the function of the hypothalamus. Hypothalamus includes neurons that secrete hormones. One of them is the gonadotropin releasing hormone, GnRH. This hormone, the gonadotropin releasing hormone, GnRH, travel to the pituitary gland through the bloodstream. What is the pituitary gland? Pituitary gland is a small gland that's connected to the hypothalamus through the hypophyseal vascular duct. The hypothalamus is connected to the pituitary gland through the hypophyseal vascular duct. Pituitary gland is composed of two parts: posterior pituitary gland, and is known by post-hypophysis, and the anterior pituitary gland. This figure is showing the hypothalamus. This is the hypothalamus, which is connected to the pituitary gland. This is the anterior pituitary, and this is the posterior pituitary region. The anterior pituitary gland secretes the gonadotropin stimulating hormones like FSH, follicular stimulating hormone, and LH, luteinizing hormone. So the anterior pituitary gland secretes the gonadotropin stimulating hormones, and we will know why it's called gonadotropin stimulating hormones, FSH and LH. Let's now talk about the pituitary control of the ovarian secretions. Experiment performed on animals allowed us to verify that the hormonal activity is not autonomous. Let's start first with the first experimental condition. We have adult control animal. It has the anterior pituitary. So we have cyclic activity of the genital system. We have cyclic activity of the ovary, the uterus, and the vagina. Okay, so second experiment, this one is the control one. This is the control experiment. This is used to compare and explain the different experimental results. Second experiment, we have the removal of the anterior pituitary gland. So we remove the anterior pituitary gland. What happens? We have atrophy of the ovaries and the inhibition of the cyclic activity. So this means that it has a role in the development of the ovaries and the cyclic activities. So here leads to the atrophy of the ovaries, and it hinders the ovarian activity, or it inhibits the ovarian activity. Whereas in the third experiment, we have removal of the anterior pituitary, then grafting it in the pituitary area. So the grafting in the area of the pituitary gland, it has the restoration of the ovarian cyclic activity, meaning that it re-established. So here we can conclude that the anterior pituitary gland, it controls the development of the cyclic activity of the ovaries. This control is ensured by blood through the release of hormones that are secreted by this. Okay? The anterior pituitary hormones FSH and LH, they stimulate the growth and the maturation of the cavitary follicle. Also, LH, the luteinizing hormone, promotes the transformation of the degenerated follicle into the corpus luteum, or the yellow. So this figure is showing the levels of LH and FSH. As you see here at the beginning of the follicular phase at day 0, this is the follicular phase. At the beginning of the follicular phase at day 0, the level of secreted FSH is approximately 4. This is the FSH, the one in red, and LH, the one in green. So the level of FSH is relatively high. It's about 4 mu per milliliter. Okay? while that of the LH is relatively low. It's about 2 mu at the beginning. During the follicular phase between the day 0 to the day 9, okay, what happens? The level of LH, it fluctuates around 2, 3, or 4 mu, as you see in this region. Okay, while between days 0 to 12, the level of secreted FSH, it decreases from 4 to 2 mu. So the level, it decreases. Around the day of ovulation, what happened? Which day of ovulation? The day 14. What happens? We have simultaneous increase in the level of LH and FSH, where LH and FSH, they increase, where FSH increases to reach about 8 mu, and that of the LH increases to reach about 24 mu. While during the luteal phase, meaning the post-ovulatory phase after ovulation in this region, both levels of LH and FSH, they decrease. FSH decreases to 2 mu approximately, and the LH decreases to the 0.1 or 0.2 mu at day 28. So here you have to know that the follicular phase, it seems to be controlled by the hypophyseal hormones, the LH and the FSH, luteinizing hormone and the follicular stimulating hormones. This increase, this control for the follicular phase will ensure the development of the follicle, stimulates the secretion of estrogen as seen here, and around day 14 we have a peak in the secretion of LH and FSH. This will allow the ovulation. Luteal phase is also controlled by the pituitary gland, is characterized by low levels of both LH and FSH. So this phase, characterized by low level of LH and FSH, will result in high levels of estrogen and progesterone during the luteal phase. So the variation in the level of the gonadotropic hormones LH and FSH are cyclic, such like the variation in the level of the ovarian hormones. So here we have something called periodicity or synchronization. Hypothalamic control of the pituitary secretions. The experiment below were carried out to show that the secretion of the pituitary hormones is not autonomous like the ovarian secretions. Let's start with the first series of the experiment. We have destruction of some hypothalamic neurons. So destruction of some hypothalamic neurons, it leads to the stop of the secretion of LH and FSH by the anterior pituitary. So we have destruction of some neurons in the hypothalamus. This leads to the inhibition or the stop of the release of LH and FSH. What does this mean? This means that these neurons are responsible for the secretion of the hormones in LH and FSH. Whereas in the second experiment, number 2, we have electric stimulations of these neurons. So you have the secretion of LH and FSH by the anterior pituitary gland will increase, and this confirms the previous result that the neurons, they are responsible for the secretion of LH and FSH. Whereas number 3, we have disconnection of the pituitary gland from the hypothalamus. So we disconnect the pituitary gland from the hypothalamus. What will happen? The anterior pituitary gland stops secreting LH and FSH. So this means that the control that's exerted by the hypothalamic neurons on the pituitary cell, it passes through the pituitary system. They should be connected. While after taking a blood sample in experiment 4 from the vascular network of the pituitary system, we have a highly active substance, which is the GnRH, may be isolating, stimulating therefore the release of the gonadotropic hormones. This means that the control between them is a chemical control, where hypothalamic neurons release into the blood the substance which is called the GnRH, gonadotropin releasing hormone. This substance or this hormone will stimulate the secretion of LH and FSH. So as a conclusion, hypothalamus controls the activity of the pituitary glands through the secretion of the hormone which is GnRH, which is released into the blood. So as a conclusion, hypothalamus will control the activity of the anterior pituitary glands through the secretion of GnRH into the blood. Some of them they ask, for example, why do we consider GnRH as a neurohormone? This is an important note. Neurohormone, since it's secreted by hypothalamic neurons and poured into the blood, for example, secreted by the neurons, it's a neuro, and it's poured into the blood, so it's a hormone. That's why it's called a neurohormone. Second series of experiment we have in the female monkey, we carry on a selective lesion in the posterior part of the hypothalamus. So before the lesion, the level of LH was about 30 microgram per liter, as shown in document 1 here. So the level of LH, it was 30 microgram per liter. And that of the FSH, it was about 350, approximately 350 microgram per liter, and the level of FSH. Upon the lesion on the posterior part of the hypothalamus, what happens? Between days 0 and day 1, what will happen? We have a falling in the level of the LH, where it falls from 30 to approximately 0, while we have a decrease in the level of FSH between days 0 to 2. In the level of FSH, which decreased from 350 to approximately 50 microgram per liter. So, as time proceeds, this level will become null at day 6 in both of them. So, how can we explain this? This can be explained that the control exerted by the anterior pituitary glands by the hypothalamic neurons is indispensable for the secretion of LH and FSH. No way to control this unless we have a control by the hypothalamic neurons. So we have a lesion, so there is no control. The level of LH and FSH, it decreases. In another female monkey subjected to the similar lesion and then injected with GnRH, either in a continuous or a discontinuous manner. So upon the lesion of the hypothalamus, what will happen? There were minimum level of LH and FSH. As you see here, the green is for LH and the red one is for the FSH. And here we have continuous injection, which is in yellow regions, and the discontinuous injection, they are in the white region here. So what can we deduce here? That upon the lesion, we have minimum levels of LH and FSH. This lesion may be corrected by the secretion, the injection of GnRH in a discontinuous manner, where the level of LH it increases and that of FSH increases also. While when we inject GnRH in the continuous manner, the level of LH and FSH it decreases again. This means that the GnRH is secreted in a discontinuous manner and not a continuous manner. It's called a pulsatile manner. Therefore, we can deduce that the pituitary gland secretion of LH and FSH is controlled, sure, by the hypothalamus that secretes GnRH. So hypothalamus secretes GnRH that stimulates the anterior pituitary gland to secrete LH and FSH. So this secretion, it should be in a discontinuous manner, in a pulsatile manner, in order to help the secretion of LH and FSH. Now we move to chapter 15, document 4, the ovarian feedback control on the hypothalamo-pituitary axis. Moving to the feedback mechanism through the experimental studies. Several experiments carried out on mammals will illustrate the effect of ovarian hormones and the secretion of gonadotropic hormones and the release of the GnRH, gonadotropin releasing hormones. So, in a female rat, a bilateral ovarectomy is done, and this bilateral ovarectomy induces a fall in the blood level of estrogen as well as an increase in the blood levels of FSH and LH. So, after bilateral ovarectomy in the female rat, the blood levels of estradiol decreases, while those of LH and FSH increases. While in the female sheep, the injection of estradiol stops. Within a few hours, the release of GnRH and leads to a decrease in LH level. So after the injection of estradiol in the female sheep, the release of GnRH stops. Okay? And this indicates that the ovary, through the secretions, through the ovarian secretion, it has a feedback, which is a negative feedback control on the hypothalamic pituitary axis. This action occurs in an antagonistic manner, meaning that in a negative feedback mechanism. In a female monkey here that was subjected to ovarectomy, we inject estradiol at time 0. Starting at time 0, the injection is continuous and constant, about 60 picogram per milliliter. When reaching time 1, the injection is unique and overdose. It reached 600 picogram per milliliter. So here the results are shown on document A, and we will talk about document A when we come to this document in the next slide. Feedback mechanisms during the cycle. The quantity of pituitary hormones secreted during the menstrual cycle is regulated through a feedback mechanism of control. Therefore, you have to know that the ovarian activity is governed by the pituitary gland, will exert activity on the pituitary gland. At the beginning of the follicular phase, when the estrogen level is low, the level of FSH increases. So low, the other one will increase. It's called a negative feedback. Next, the moderate level of estrogen provokes a decrease in the level of FSH. So moderate level decrease. So one of them increase, and as it decreases, it's called a negative feedback control. At the end of the follicular phase, the estrogen level increases, and this exerts a positive feedback on the hypothalamic pituitary. This will induce a secretion of GnRH that, in its return, will stimulate the release of LH. It's called pre-ovulatory surge, as well as a small peak of FSH. During a luteal phase, high levels of estrogen and progesterone through their secretion of the yellow body exerts a negative feedback control on the hypothalamic pituitary axis, and therefore on the release of gonadotropic hormones, LH and FSH. So this document A is showing the effect of the injection of estradiol on the evolution of the LH level. So after the female monkey was subjected to ovarectomy, there was no secretion of estradiol before time 0, okay, as shown here. After time 0, this indicates that before we move after injection at time 0, this indicates that the ovaries, they are the main source of estradiol. Since after time 0, there is no injection of estradiol, there is no secretion of estradiol, and we have ovarectomy, so no estradiol, so it indicates that the ovaries are the main source of estradiol. Before T0, the level of LH, they are higher. They are about 26 approximately, or 27 microgram per milliliter. And this confirms the noted about the antagonistic manner of the secretion of estradiol and LH. This is high and this will be low. Okay, this is antagonistic manner or a negative feedback control. From times T0 to T1, we're talking about this region. Time 0, T0 to T1. What happens? After continuous and constant injections of low levels of the estradiol, levels of estrogens, they are maintained, or estradiol, they are maintained about 60 picogram per milliliter. While that, the LH, it decreases. While at time T1, what will happen? we inject a unique overdose of estradiol, about 600 picogram per milliliter. What will happen here? The level of LH, it increases to the maximum to about 37 to 36 nanogram per milliliter. So after time t1, the level of estradiol will fall, and similarly the level of LH will go back to the normal level. This means that, and pay attention please, this means that the secretion of LH depends on the doses of the estrogen or an estradiol injected. This is ensured either by a positive feedback, as shown here, both of them increase, or by a negative feedback control. When the level of estradiol is moderate, what will happen? This inhibits the secretion of LH. So when the level of estradiol is moderate, we have inhibition in the secretion of LH. This is called a negative feedback mechanism. Whereas when the level of estradiol is high level of estradiol, this exerts a positive feedback, and this will lead to the increase in the level of LH as shown in this figure. So here, as I said, document D showing the regulatory system of the ovarian hormones, you have to know that when the level of estradiol is moderate, this is document C and D, the feedback mechanism. When the level of estradiol is moderate, so this will inhibit the secretion of LH, and this is called a negative feedback. Minus meaning that decrease in the secretion, so decrease in the secretion of LH. While when the level of estradiol is high, so high level will exert a positive feedback on the hypothalamus and on the anterior pituitary gland, and this will lead to the positive feedback control. And regulatory system and the ovarian hormones, you have to know that blood levels of ovarian hormones reference value. So you have to know that first, hypothalamus pituitary system detector and integration system center secretes LH and FSH, and this will regulate the system through the feedback controls, either the positive feedback control or the negative feedback control. So you have to know here that the regulatory system of the ovarian hormones consists of regulated system, regulating system, and feedback control circuit. Regulated system corresponds to the parameter ovarian hormones that triggers the regulatory mechanism when its values deviate from a standard or a reference value. This regulated system corresponds to the blood level of the ovarian hormones. Regulated system is composed of three successive elements: a detector consisting of a receptor that compares the value of the parameter to that of the reference value, integration center that responds to the deviation message by emitting an outgoing message, which is a correctional order. The detector and the integration center, both they correspond to the hypothalamic pituitary system. Correctional system. This correctional system, it has an important role. It's constituted of one or several organs or effectors. They are able of bringing the disrupted parameter back to its original reference. This correctional system is correspond to the ovary. Right, the last one, we have the feedback control mechanism, which allows the regulation of the level of the hypothalamic and the pituitary hormones either through a positive feedback or through a negative feedback control.